In this short video today, we'll take a look at the structure of DNA and how this can be analysed by a particular process called gel electrophoresis to give us all our unique genetic barcodes. This should also help you solve the activities on the TrioSci website. Here's a reminder of our common cell types, and if you've watched my previous video on cells, you should recognise this image. We know the nucleus is the control centre of our cells, containing information in the form of DNA. Today we'll explore DNA and one particular method of analysing it. Let's start this journey with a recap of the nucleus. So it's the largest organelle, accounting for about 10% of the volume of a cell, and we already know it controls the cell's activities and contains some sort of genetic information which acts as instructions for the cell. Now let's dig a little deeper and see how this genetic information is stored in a special structure called chromosomes. Here you can see an enlarged image of what chromosomes look like inside a cell's nucleus. They actually occur in pairs, 23 pairs in fact, and since we inherit one from our mother and one from our father, we actually have 46 individual chromosomes in total. Chromosomes are long threads of DNA wrapped around small balls of proteins or histones to form nucleosomes. This helps them compact tightly together so we can fit in all the important information, since a human cell contains a whopping 3 meters of DNA. But what actually is DNA? It's a large, complex polymer. You might have heard of this term before in chemistry. It means a molecule made up of lots of small repeating units, or monomers. And we've mentioned our nucleus provides some sort of instructions to the cell. Well, this is actually called the genetic code, a list of instructions, much like a recipe, which determines the characteristics of an organism. For example, eye colour, hair colour, height, or skin tone. And everybody's DNA is unique, with the exception of identical twins, which is why we're all very different to each other. But how was DNA discovered? In the early 1950s, American biologist James Watson and British physicist Francis Crick came up with their famous model of the structure of DNA. They were awarded the Nobel Prize in 1962 along with another scientist called Maurice Wilkins. Can you go away and work out what he contributed? So they mainly used existing data and information and pieced it together in a different way to be the first to discover this structure. Their work heavily relied on findings by Rosalind Franklin, who was not recognised for her contribution until after she died. She was an expert in a process called X-ray crystallography, exposing crystallised molecules such as DNA to X-rays, which deflects the rays in a specific pattern and gives information about the molecule's shape. They used this X-ray information to suggest the double helix structure of DNA, 2 strands of DNA called together to form a double helix. This actually has very specific characteristics. Go and hop onto Google and see if you can find out what these are. Now let's take a closer look at the structure of DNA which Watson and Crick discovered. We know it's double-stranded, comprising of 2 strands intertwined to form a double helix. Quite simply, each strand is made up of bases and a backbone. Let's take a look at the bases first. The human genome is made of about 3.2 billion bases, but this can vary for each organism. There are 4 bases, adenine, cytosine, thymine and guanine, which can be simplified to the letters A, C, T and G, and act a little bit like an alphabet. But how do these 2 single strands of DNA stay together in the double helix formation? Well, they're connected by hydrogen bonds between the oxygen and hydrogen of each of these bases. This creates a ladder-like structure and causes twisting, giving the DNA its distinctive double helix shape. You can see the bonds here in more detail. Now let's take a look at the backbone. Here's a small section of our backbone, or sugar phosphate backbone to be specific. So why is it called this? Well, the backbone comprises 3 important sections: a phosphate group, a sugar group, and our base. This can be any base from our alphabet, and the random nature of this is what gives each of us our unique genetic codes. If you look closely, you may be able to see a small negative sign on the phosphate group. This means it's negatively charged, as it has spare electrons, and hence gives DNA its overall negative charge. We'll revisit this a little later. So what's this genetic code we've been talking about? Here you can see how the order of bases along a single strand of DNA looks a little bit like a secret code, or in this case secret instructions. Now this is decoded by exiting the nucleus, so it can be read by specialist machinery in the cell's cytoplasm. But to be small enough to exit the nucleus, our DNA is converted into a small messenger molecule called mRNA. This secret code can now be read by the cell's machinery. 3 adjacent bases are read as a 3-letter word, or codon, and code for one of 22 amino acids. These amino acids are the building blocks of our proteins and fold many times to create highly complex and organised 3D structures, which we call proteins. So this sequence of amino acids, and therefore proteins, can be divided into genes. These are sections of DNA which code for a specific protein. A gene may consist of 4 or 54 amino acids, and each gene has its own characteristic, such as eye colour or hair colour. These genes act as discrete units of heredity and can be passed on to the next generation. Now, an allele is a particular form of a gene. For example, the gene for fur colour has an allele for white fur and an allele for black fur. A rabbit may have 2 of the same alleles, known as homozygous, for white fur, in which case it will be white, or it may have 2 different ones, called heterozygous, in which case its fur colour would be decided by the dominant allele. But that's a lesson for another day. Give it a little Google if you're interested. Let's quickly go back to this image we saw earlier. We can now be a little more specific, and instead of saying just DNA, we can call small sections of the genetic code genes. These genes can be separated by special enzymes and broken down into fragments, with each of these fragments being a different length. So by fragmenting or breaking down a sample of DNA and separating these fragments by size, we can create an individual characteristic profile of DNA bands for each individual, which looks a little bit like a barcode. There's a couple of scenarios where we can use this barcode to identify individuals. For example, if a baby is born and the mother doesn't know who the father is. Now the child will have half its mother's DNA and half the father's DNA, so from this we can work out who the father is. Or in forensic science, the criminal's DNA is often left behind at a crime scene. If the police have a list of potential suspects, they can sample their DNA and compare this to that left at the crime scene to work out who was the criminal. For this, we use a technique called gel electrophoresis. This method separates DNA fragments according to their size to give a unique barcode for each individual. Let's take a look at how it's done. Here, we have a slab of jelly, or agarose gel, which DNA can move through. And at one end, we have a couple of small wells which we can load our DNA samples into. Now we hook this up to a power source, and since DNA is negatively charged, remember how we talked about the negatively charged phosphate groups earlier, it travels towards the positive end of the power source. And our agarose jelly acts a little bit like a sieve, allowing small pieces to pass through easily, but trapping the larger fragments. So the smaller pieces of DNA travel the furthest, and the larger pieces get stuck behind. We can measure the size of these fragments using control ladders. Here we have the barcode, or genetic profile, for a DNA sample found at a crime scene. The police have identified 4 suspects and taken DNA samples from each of them. When we run this gel, we'll be able to compare the genetic profile of the suspects to the crime scene to find out who is guilty. Can you work out which suspect is the criminal? Take a minute to have a look. I'll give you a clue. Their barcode should match that found at the crime scene. Did you guess correctly? The correct answer is suspect D. But it's not only humans which have a unique genetic profile. Every species does, including plants. Do you remember looking at pollen grains from different species of plants? These 3 plants can be distinguished by the shape of their pollen grains. But not only does their pollen look different, each of these plants also have their very own genetic profiles. Can you recall the types of plant these pollen grains belong to? Here we have clover, dandelion, and bluebell. So we can carry out a similar experiment to the one we just did at a crime scene, but this time analyse samples from each of our plants to work out where our mystery pollen comes from. Here's a gel electrophoresis experiment using DNA samples from each of our plant species, clover, dandelion and bluebell. Can you work out which species our mystery DNA sample is from? Now you can use what you've learnt here to complete the activity on the TrioSci website. But I'll give you a quick hint, the answer might not be the same. If you fancy a challenge, give this one a go. Here we have electrophoresis results from a mother and child, but we're not sure who's the father. Can you use this information to work out which of the three potential dads is the child's actual father? Now remember, you receive half your DNA from your mum and half from your dad, so the child's genetic profile will be a combination of both parents. Thanks for watching. We hope this video helped you understand why gel electrophoresis is a super easy way of analysing DNA and why it can be really useful in the real world. Keep an eye out on the website for more videos and activities coming soon.